Here in chapter 6, we are going to be talking about microbial genetics, including the structure of DNA that applies to all living organisms, how that DNA gets replicated, how it's used to make proteins, and then also talking about how bacteria can go through that horizontal gene transfer that we've talked a little bit about already. So first of all, some terminology. Genome. This is the sum total of all the genetic information in the organism. So this includes the DNA and RNA. For the most part, this exists as chromosomes, but there are some non-chromosomal forms. So for example, we've talked about plasmids before that contain extra pieces of DNA. They are, if we see the picture over here, they're just, again, not part of the main chromosome. They're not required for the organism, but they provide some type of benefit. So for example, bacteria have one chromosome that contains all their needed DNA, but they can also have these extra plasmids. And then in eukaryotic cells, organelles, also some of them have DNA. So mitochondria and chloroplasts, for example, have some DNA within them. So that would be part of the genome as well. So again, it's all of the DNA that's in there. And then even viruses can do that as well. So again, genomes of cells, we're talking about all the DNA, all that genetic material within the organism. Viruses, though, can have either DNA or RNA. They don't have both, but their genome would be one or the other. And then the term genomics is just the study of the entire genome. So how all that DNA works together and how it creates the characteristics for the organism. So some of the levels of that genome. So again, specifically talking here about DNA. So we mentioned chromosome already. So this is just a neatly packaged DNA molecule. So each chromosome has one DNA molecule. It's extremely long, but it's, again, each DNA molecule is packaged onto one chromosome. Bacteria cells have one chromosome, and that's it. Us, for example, we have 46 chromosomes in each cell, just to kind of put it into perspective. So we have 46 DNA molecules, each packaged into a chromosome. Now, while both of them have chromosomes, eukaryotic chromosomes, those are found in the nucleus, and they have various numbers. So there can only be a few of them. Some organisms have hundreds of different chromosomes. And they can be in pairs, and that makes up a cell called a diploid cell. That's what our cells are. We have 23 pairs of chromosomes, or they can be haploid. So our egg or sperm cells, for example, only have one of each chromosome. And they are also linear, so they're found in straight lines. Bacterial chromosomes, though, again, there is only one single chromosome, and it is circular instead of linear. But again, they can also have some others as well, but for the most part, all of them, again, there's some, of course, exceptions to the rules, but for the most part, single circular chromosomes. Now, the gene, then more terminology here. This is essentially a packet of information in each chromosome. So if we take a long chromosome made up of lots of DNA, that DNA gets packaged into these little packets called genes. So again, it's subdivided into these, and each gene has a code to provide a function. For the most part, they're each going to be making different proteins. So we'll be talking about transcription and translation and how that happens. But some of the genes are also used to make RNA that then makes the protein. But then some of them are also regulatory. So they're not actually making a protein, but they're controlling whether the genes get turned on or turned off. So again, we have structural genes, we have genes that directly lead to protein production, and we have regulatory genes. Now, genotype versus phenotype, a couple more terms. Genotype, again, is the sum of all the genes that make up the organisms. So again, we have the genome, which is all the DNA. Genotype is the sum of all the types of those genes. And then phenotype is actually the expression of the genotype. So our cells have lots, and same with the bacteria, they have lots of different genes, but they don't use all of them, or they don't use all of them at the same time. So their cells have ways of expressing or turning on a gene to make a protein or to regulate something, or they have the ability to turn it off. So the phenotype is actually the expression using those genes to create a specific function or trait. So like hair color, eye color for a bacteria, whether or not it has a capsule or whether or not it has specific receptors or a flagella, that's the phenotype. So all the organisms contain way more genes in the genotype than they express. So again, not all of them actually get used or turned on either at the same time or sometimes even within the organism's lifetime. Now, if we look a little bit at the structure of DNA, and I'm sure you've covered this before, so we'll just kind of highlight some of it. So first of all, the basic unit of DNA is called a nucleotide. And a nucleotide is made up of 3 parts. It has a phosphate group. It has a deoxyribose sugar. That's where the name deoxyribonucleic acid, which is what DNA stands for, comes from. And then it has what's called a nitrogenous base. And those bases can either be purines or pyrimidines, and we'll see a picture here in a minute. Purines are adenine and guanine, and the pyrimidines are thymine and cytosine. And DNA also exhibits base pairing to where A always pairs with T and G always pairs with C. So we say that they are complementary. Now the DNA also has an anti-parallel arrangement because it's double-stranded. It has 2 strands of these nucleotides. So one of the strands runs in the opposite direction of the other. So each nucleotide strand has a 5' and a 3' end. So one of those strands goes in the 5 to 3' direction. The other strand goes in the opposite, 3 to 5. So they're anti-parallel. And then there's a significant factor in DNA synthesis and protein production. So that's what the DNA is predominantly used for, to make more DNA to pass on to offspring and to make proteins for that phenotype. Now here we can see the actual structure of the DNA. So again, each nucleotide, just highlight one of them, has a phosphate. It has this deoxyribose sugar, and then it has a base. So one of those nitrogenous bases. Again, just moving down here, we can see that adenine and guanine are both a little bit bigger. They have 2 of these little ring structures, and those are purines. They're a little bit bigger. And then thymine and cytosine, they are the pyrimidines. They only have a single base or a single ring structure. So let me get rid of this real quick. And then that base pairing that we mentioned, again, adenine and thymine always pair together, and guanine and cytosine. So again, we say the strands are complementary. If we know the sequence on one side, so T, G, C, and A, using those base pair rules, we know the opposite strand would be A, C, G, and T, because that's the way those bases line up. Get rid of this here real quick. All right, so then the last thing here with the DNA is that it is anti-parallel. So the strands are directional. The phosphate group makes up what's called the 5' end that has to deal with the sugar or the carbon that it's attached to. And then the sugar unit is the 3' end. So here we have the phosphate at the top and the sugar at the bottom. So we say this is going in the 5 to 3' direction. So then the complementary strand it's attached to is going in the opposite direction. So the 3' end is here and the 5' end is at the bottom. So again, our phosphate is starting at the bottom, and then our 3' end here has that sugar unit. So it's going in the 5 to 3' direction. Now, ultimately, those strands twist to form this helical structure. So we say that it is double-stranded. It has 2 strands of nucleotides, and they twist into a helical structure. Now, in order for organisms to reproduce, they have to be able to copy their DNA so that the offspring or the other cells receive a copy of the DNA. So that process is called replication. And for all organisms, whether they're prokaryotic or eukaryotic, replication is what we call semi-conservative. And that means that each daughter molecule that's made, so the two DNA molecules at the end, are identical to each other and they're identical to the parent. Now, neither of them is completely new, so that's what we're saying, semi-conservative. So one of the strands, one of those nucleotide strands is called the template. It's the original strand, so it's conserved. The other strand gets synthesized, so it's new. So at the end, each DNA molecule has one strand of DNA that was old, it was original, and one new strand that was just made complementary to that template. So if we look here, we can see this. Again, we're focusing on bacteria. For this, it's a little bit different in eukaryotic cells. But at either place, they have an origin. So replication origin, where it's starting, as the name there implies, where replication would start. And essentially what happens is these two strands shown in blue here, this is the original DNA. Those are going to be separated by an enzyme here. It just essentially unzips the DNA. So then one template strand goes off to the side and the other one goes to the other side. So it's just separating those hydrogen bonds, breaking them up. And then, so these are going to serve as our old strands. So here in blue, let me just write. So here in blue, this is our old strand. It was original. We didn't make this. We just separated it at the beginning. Then enzymes are going to use those base pairs. So if there's an A on the original strand or the template, it puts a T. If there's a C, it puts a G, and so on. So we are making a new strand of DNA that's complementary to the old strand. So by the time this is complete, that's hard to write. So by the time that replication is complete, we end up with two DNA molecules. We started with one, and each one has an old or original strand, and then one new strand that was just made. And so it's called semi-conservative. Now, this DNA not only is used to make more DNA, but it's also used to make proteins to determine that phenotype. And the way that it does that follows what's called the central dogma. And this is a two-step or two-stage process. Each stage has multiple steps. But that DNA is first used in a process called transcription to make RNA. Then in the second stage, that RNA in translation is used to actually make the protein. So it's kind of a two-phase process. DNA is used to make RNA, then that RNA can be used to actually make the protein. So it's now not all of the DNA is used to make protein, so it's not always complete. So it depends on the purpose. So some of the RNA molecules that get made in transcription are not used to directly make protein. Instead, they regulate gene function. So they are a product of their own. So the end result in that case is just the RNA. It doesn't get translated to protein. And it's actually been found that a lot of genetic malfunctions, so different diseases, are due to changes in the regulatory RNA, not the protein itself. So it's a mutation or a change in the RNA. And again, it's an RNA not used to make a protein. And then also there is DNA that codes for crucial RNA that was once thought to be junk. So we knew that there was all this kind of extra DNA, and it was determined to be, useless or junk. But as more and more information is becoming available, we're finding that those RNA molecules play a role in regulation, or they have other functions besides just making protein. All right, so just kind of the summary here of this central dogma. So again, we have our DNA that has our instructions. Again, each one has an old strand and a new strand from that semi-conservative replication. So again, during transcription, one gene, so not the whole DNA molecule, but one gene at a time, is transcribed from DNA into RNA. And there are multiple types of RNA, and we're learning more about other types as well. But the three that are needed for translation is tRNA. It goes and gets amino acids. The messenger RNA that actually has the information to make the protein, the instructions. And then we also have ribosomal RNA, and that helps make the ribosomes, which is where translation happens. So once we have our messenger RNA, then during translation, that messenger RNA is used by the ribosome. So again, that's where protein synthesis happens. That's why antibiotics that inhibit ribosomes are so effective, because the bacteria can't make their proteins that they need. So that messenger RNA is attached to the ribosome, and using the tRNA and the ribosomal RNA that makes up the ribosome, that information from the mRNA is used to bring amino acids together to make a specific protein. So we have transcription and then translation. Now, again, there are also many genes in our genome that are not used to make messenger RNA to make a protein. Instead, they are regulatory RNAs, and these include microRNA, interfering, antisense, what are called ribose switches. And again, we're learning more and more about these additional RNA molecules and how they impact the genome. So this gene to protein connection, so the connection between the DNA and its traits, again, genotype to phenotype. So the protein's primary structure, which is the amino acid sequence that it has, ultimately determines the shape and function of that protein. So again, those proteins then determine the phenotype. And the study of that is called proteomics. So we have genomics that studies all the genes. Proteomics studies all of the proteins that are expressed in an organism. So DNA is mainly the blueprint then that tells the proteins how much to make and what type of proteins to make. And this is showing the simplified version of that central dogma. So the DNA, again, double-stranded here. Only one of those strands, in this case the bottom strand here of DNA, is used. The other one is completely ignored. That one strand of DNA is used to make messenger RNA, so using those base pairings, so C pairs to G, G to C. The exception, though, is that RNA has uracil instead of thymine. So if there's an A on DNA, it brings in a U in RNA. But ultimately, those get transcribed into RNA nucleotides, and then they're broken up into groups of 3. And each group of 3 is called a codon. And each codon brings in one amino acid. So that messenger RNA is broken up into groups of 3, into codons, to bring in individual amino acids. And those amino acid sequences ultimately make those proteins. For transcription and translation to happen, there are several key parts. We already mentioned these, but again, messenger RNA, that's going to actually carry the DNA information, the instructions to make the protein. The transfer RNA, that's actually bringing the amino acid that would be attached here to the ribosome so that it comes to the right code. The regulatory RNA and the ribosomes and the ribosomal RNA that make the place for translation to happen. Lots of enzymes that are there, and raw materials, like we need phosphates and we need amino acids, and we need sugars to make all of this happen. All right, this just highlights and summarizes the different types of RNA. So again, we have messenger RNA. That is actually transporting that code to the ribosome. It does get translated into protein, but it's the only one. All of these other RNA molecules, they are the final product. So there's just transcription to make them, and then they have a function. So again, transfer RNA, that's bringing the amino acid. Ribosomal RNA, that makes the ribosome. And then we have these other regulatory RNAs like microRNA and ribose switches. And as the name implies, they regulate that gene expression. So they turn on or turn off genes. We also need primers to actually get the DNA ready so that it can be transcribed. And then for eukaryotic cells, we need ribozymes and spliceosomes that remove what are called introns extra DNA molecules before translation happens. So then RNA, again, just a little structural difference to DNA. So DNA is deoxyribonucleic acid. RNA is ribonucleic acid. And there are a few key differences between DNA and RNA. So RNA is single-stranded, so it's one nucleotide strand. So it's not double-stranded. There can still be some base pairing that lets it kind of twist and loop on itself to give it some different levels of complexity. But again, it's one strand of nucleotides. As I mentioned before, it has uracil instead of thymine. So if there's an adenine, then it's going to pair with uracil. And then that sugar, which is where the name comes from, is ribose instead of deoxyribose. So the messenger RNA here, again, that's the transcript of the gene from the DNA, and it is used, it gets made during DNA replication, but that actually gets used to make the protein. So again, that messenger RNA gets divided into groups of 3, so triplet bases, and that's going to bring in 1 amino acid. So it gets divided into groups of 3, which are called codons. Now the transfer RNA, again, has the sequences, has those hydrogen bonds, those complementary, again, A would pair to U, C to G, G to C. So it has kind of a 3-dimensional shape. Now it has a couple parts. So one, it attaches to the amino acid, but it also has what's called the anticodon. So again, the codon is a group of 3 nucleotides on RNA that brings in an amino acid. Well, the anticodon is found on the tRNA, and it complements the messenger RNA. So it would go along with it. That's how we match up the tRNA to the messenger RNA. So the right amino acid is coming in, the anticodon attaching to the codon. A little bit more about the ribosome. So again, ribosomal RNA is used to make this. So again, 3-dimensional shapes. There are 2 subunits to the ribosome, and again, this is where we get the different sizes between prokaryotic and eukaryotic ribosomes and why they're good targets for antibiotics. So again, there's slightly different units, which are S. So we have 50S and 30S's. S is just the unit for measurement for these. But again, 2 of those subunits, a smaller and a larger, come together to actually get translation. So here is the small ribosomal subunit. Down here at the bottom, that's where the messenger RNA attaches, and then the large ribosomal subunit attaches once the messenger RNA is there to complete the ribosome, again, made up of those structural RNA molecules and some other proteins as well. All right, so if we start talking with transcription and how it happens, so first of all, initiation, so how does it start? So with these, there's what's called a promoter region. So on the DNA, there is this region called the promoter. And it's a sequence of A's and U's and all of that that come together that tell the enzyme RNA polymerase where to start. So it's at the beginning of the gene, RNA polymerase attaches, and that's going to start transcription. So the binding of RNA polymerase to the promoter on DNA. Then the DNA molecules separate, so those hydrogen bonds separate, and then a single strand of the DNA is used. That other strand is completely ignored. It's just down here hanging out. So we separate the 2 strands, similar to replication. But what's happening here is RNA then is being made. So we use those complementary sequences, A pairs to U, C to G, etc., making this RNA molecule. Now again, RNA is single-stranded, so as the RNA falls off, then those 2 DNA strands go right back together. So they're zipping back as the RNA polymerase moves along, and that's referred to as elongation. And that's going to continue until we reach the final stage, which is termination, which ends it. And this is where it gets to a terminator sequence or a DNA sequence that signals termination. So the promoter starts it. That's where RNA polymerase knows where to start. It separates the 2 strands and adds the complementary RNA strands to make the RNA. And then it reaches another sequence at the end of the gene called the terminator, which ends translation, or sorry, which ends transcription. All right, so this master genetic code, so the principles then of translation, so that messenger RNA, again, is grouped into groups of 3 called codons, and that dictates which amino acid comes in using that anticodon. So again, except for a few cases that code is universal. So we know for every group of codons which amino acids that's going to bring in. And that is shown here in what's called the genetic code. So for every possible codon, this shows which amino acid would be brought in. And again, it's rather universal. It doesn't matter if it's prokaryotic, eukaryotic, or viral. Whenever we have a CUU codon on that messenger RNA, the tRNA will bring in a leucine. If there's a UCU, then it brings in a serine. If there is a GAU, the tRNA brings in aspartic acid. So using that anticodon on the tRNA, it would recognize this GAU, and attached to that tRNA would be the amino acid aspartic acid. All right, so we can use this genetic code. If we know the sequence of RNA, we can then determine the sequence of amino acids. Now there is one start codon, as the name implies, that will start translation, as we'll see here in a little bit. It also brings in the amino acid methionine. And then there are 3 different stop codons. So UAA, UAG, and UGA, and that will signal the end of translation. So how do we interpret the code? So again, here is our DNA molecule. So again, one of those strands will be used. So in this case, we're using this bottom strand of DNA. So again, the promoter would be here at the beginning. RNA polymerase would bind. We would separate the 2 strands, and using just this strand, which is called the template strand, that polymerase molecule would make complementary bases. So again, A would pair to U, or sorry, T would pair to A, the A on DNA would pair to U, C on DNA would bring in a G, G would bring C, and so we just keep going down using those base pairs, so that RNA polymerase would be bringing in these RNA molecules. Then those RNA molecules get grouped into groups of 3. So to make our codons, we always start, as the name implies, with a start codon, which is AUG. Then using the anticodon on our tRNA, so A and U would pair, U and A would pair, G and C would pair, and using that genetic code we had just looked at, we know that the amino acid methionine would be attached to that tRNA. Then the codon CUG, the anticodon of the tRNA, C would pair to G, U to A, G to C. And if we use that genetic code, we know that the amino acid it would bring in is leucine. And again, going on down until we reach one of those stop codons. So for translation, again, this is the second part then of that gene expression. So transcription makes the RNA, translation actually makes the protein. So we need, of course, the message. We need the tRNA to bring the amino acids. We actually need the amino acids, and we need ribosomes for that place to occur. And just like with transcription, we have initiation, elongation, and then it's terminated. So if we look at some of the terms here, so start codon I'd already mentioned. This is the first 3 nucleotides on the RNA. It is always AUG. Now that may not be at the beginning of the RNA molecule, so we may have, our RNA may be A, C, C, let's say G, and then we would have an A, U, G, and then let's just make up some other stuff here. So again, our RNA molecule, these are all the nucleotides. We don't just start right here at the beginning because we have to have an AUG. So instead it kind of moves along here until it finds that AUG, and then we start grouping into groups of 3 to form those codons. And then again, just as we have a start, there are also stop, but there are 3 of those. So encountering any of these 3 would terminate or end translation. And then the term translocation, just meaning to change positions. This is the shifting of the ribosome. and it moves down one codon at a time. So if the ribosome is here, translocation would move it then to here over this set of codons, and it just keeps moving down one codon at a time. All right, so how does this actually happen? So first of all, again, our messenger RNA would attach to that small subunit. We have a start codon here, AUG. Our tRNA comes in, attaches with the anticodon, and has this amino acid methionine. Okay, our next codon is CUG. Again, tRNA comes in, binds using its anticodon, and brings leucine. Now, these two amino acids, let me erase this. These two amino acids are now right beside each other, and that facilitates the formation of what's called a peptide bond that is going to form and join those two amino acids together. Once that bond is formed, now the RNA is going to move down one codon. That's that translocation. And now what happens is we've now opened up a new codon. So we get this translocation, this moving. So now we're going to bring another amino acid in here to match up. So if we see here, so again, our first translocation, we're moving down to where now the CCG codon is here in our ribosome. Our next tRNA that has an anticodon to recognize it comes in. That brings these two amino acids close together so they can form that peptide bond. And then once that peptide bond is formed, now we're going to move down another codon, and everything just keeps shifting, and we keep bringing in more and more tRNAs to match up with those codons until we get to a stop codon, and then everything ends. Now, in bacteria, they can kind of speed this process up. So in eukaryotic cells, transcription has to happen in the nucleus. That's where the DNA is. Then the messenger RNA has to leave and go to ribosomes for translation to occur. So they're two separate events. In prokaryotic cells, though, because they don't have a nucleus, as soon as transcription starts, so here's our RNA polymerase moving along, making our RNA here in orange. Well, as soon as that RNA starts to be made, a ribosome attaches to it, and it starts translating the process. Okay, so we're starting here with our messenger RNA, that ribosome attaches, and it starts making a protein. And it's just going to keep moving along through translocation. It moves along, and then our protein is spiraling off the side. Once this ribosome moves so far, another ribosome comes in and attaches. So you can have hundreds of ribosomes attached to this messenger RNA at any given time, and that speeds up the process.